Okay, good afternoon and good evening. I would like to thank the organizers of the APC CMI for inviting me to present at this session. So today I will be speaking about arboviruses in Asia. My name is Moi and I'm from the Graduate School of Medicine, the University of Tokyo. So to begin with, our work mostly involves collaboration of infectious diseases in the Southeast Asia region. So we've been working on dengue, arboviruses, emerging, re-emerging diseases. So this is an overview of what of the research which our institute is involved in, and we are also working on disease control section for the research which involves diagnostics and also vaccination and therapeutics as well. So to move on, I was previously with the Department of Virology, the Institute of Tropical Medicine. This is in Nagasaki University. So most of the work will be based on the collaboration between this institute and also with other collaborators in the Southeast Asia region. So to begin with, I think the previous speakers have already elaborated on dengue and flaviviruses. Well, there are 4 different serotypes of the virus itself. And one of the central questions is to why do some people develop severe dengue and why does they have a tendency to develop severe dengue during secondary dengue infection? So to begin with, we are interested also in how this prior immunity towards dengue and what sort of arboviruses, specifically flaviviruses, causes all this infection and what sort of impact does it have on the subsequent infection of dengue. So from the previous vaccination lessons from dengue, it has been going on for more than 70 years. However, there are currently no effective vaccine for the disease. And what we can see here is that these antibodies towards dengue, they tend to wane, they tend to weaken after some time. And this is during when you see there's an increase in risk of severe dengue. So in this case, we have some central research questions, which includes why are there some dengue breakthroughs even in patients with neutralizing antibodies towards dengue, as well as what are the levels of these immunity or antibody that confers towards protection. So there remains a need to define what is disease protection in dengue and arboviruses in the region, as well as how does all this interplay between the epidemiology and how does all this affect the disease epidemic in the region. So what we've been looking on all these while includes several sectors, which is the host, vector, virus, and all these is important to address the challenges in vaccine and therapeutics development. So our research fieldwork includes looking at what sort of dengue is causing outbreaks, and this case is Vietnam. We see that there's an ongoing dengue 1 outbreak, which has been going on for since 2001. And we have also explored some other flaviviruses, which are causing epidemic in the region. So this was back in 2017 to 2018. And this is one of the earliest reports of microencephaly, which is associated with Zika virus in the region. This is a case report in Vietnam. And then we went on to look at how long has this Zika epidemic been going on in Vietnam? And we found that it has been going on even prior to reports of the outbreak in Vietnam. And that was approximately in 2014, in which we identified several different strains of Zika virus that has been causing outbreaks in Vietnam prior to the official reports of the epidemic in the region. So again, we went on to explore. There are also changing landscapes of dengue serotypes and genotypes in Southeast Asia region. We are also working in collaborators with Vietnam. We found that there's an increase in dengue 3 a genotype 3 outbreak in the region, and there's an introduction of new genotypes in Myanmar as well. And whereas in the Philippines, there has also been some changes in the genotypic changes in dengue 4 in the Philippines as well. So in this case, we have offered feedback to the local authorities, as well as with this, it's important to find out what exactly are the serotypes and genotypes which are causing outbreaks in the region. So the second part of the dengue epidemiology is that the dengue activity with dominant serotype and genotype replacement causes a higher number of cases each time there's a replacement of the dominant serotype or genotype. And there are also questions as to does some virus strain cause severe disease? And in this case, we did a comparison between the characteristics of the dengue infection and the virus itself in relation to the epidemic trend, serotypes, genotypes, as well as study of the virus genome sequences between several countries in Philippines, Vietnam, and Myanmar. And we also have identified several quasi-species of dengue virus and also elucidated their role in differential dengue presentation. So in this case, we have a outbreak of dengue 3, which is related to neurological involvement in Vietnam. And here we found that this particular strain of dengue virus, there are two different types of strains which were isolated from the CSF and also from the patient sample itself. So we went on to look at the growth rate of the virus, the two different isolates from a patient with neurological presentation and from another patient with dengue 3 at the same outbreak. And we found that this virus, which is isolated from the CSF, tends to grow better in neural cells. And so to summarize some of the pathogenesis studies of this dengue virus, we found that the non-structural region is most likely playing an important role from in the CSF-derived dengue 3 virus, which may cause enhanced infectivity in these neuroblastoma cells. So again, we went on to look at the host section, which is how does prior immunity, for example, Zika or subsequent dengue or other JEV flavivirus exposure cause severe presentation or vice versa. So we went on to look at so first I have to go back to introduce the AD hypothesis as to why some people may have antibodies that are cross-reactive, but these antibodies do not actually neutralize the virus. And in fact, it enhances infection and in the end leads to the production of more virus progeny, which is the hallmark of severe dengue. So in this case, during primary infection, in the absence of these cross-reactive antibodies, some possible outcome could be mild or lesser severe presentation of the dengue itself. Whereas in the secondary infection, in the homologous type, these neutralizing antibodies or these homologous antibodies could lead to neutralization of the virus, and in the end, it could lead to a possible outcome of protection. However, for heterologous secondary dengue infection, these cross-reactive antibodies do not neutralize the virus, and it in turn leads to higher production of progeny of the virus, which may lead to a possible outcome of severe dengue. So the research question here is that if you have all this pre-existing immunity to flaviviruses, does it give you better protection, or is that not the case? So we need to have an assay which determines the AD activity. So this has been done previously. We have made an assay which can detect the AD activity of the antibodies. So using this new assay, well, we found that during primary dengue infection, there's no virus immune complex that may offer higher viremia levels during as compared to secondary dengue infection. So we went on to develop a much more rapid assay of the AD assay. So this is a luciferase-based neutralization AD assay. So the previous one was a plaque-based, and in this case, it can be adapted to 96 well and a 384 wells format. And it is much more rapid as compared to the previous system. So one of my graduate students will have a presentation on this system in the poster section of the conference. So let's go a little bit back to explore on the pre-existing immunity or during primary infection, the first exposure to dengue or perhaps flavivirus. So what we see here is that there's an induction of IgG and IgM antibodies, which goes down over time. And over time, this IgG may wane, they may disappear, but not entirely disappear. And however, with the secondary infection, it may cause a stronger reaction. And in this case, you will see much more antibodies that may neutralize the virus. So the immunity is complex. We know that there's a whole host of cells which plays a role in severe presentation of dengue. But in this case, whenever we look at the antibodies, we're always looking at the levels of antibodies which are going down. But behind that, there are also a whole host of cells, for example, the CD4, CD8 T cells, and also B cells that may play a role in maintaining all these antibodies that we finally measure as AD antibodies. So we are actually interested into looking into what sort of antibodies are actually playing a role in giving you all these different disease presentation. So we are also looking into this AD peak enhancement titer, which you have a dilution of these samples, and then you can see that there's a titer which gives you a peak AD. So in this sense, we've used this to look at primary and secondary infection, and using different genotypes, or a panel of dengue-1 genotypes, we have looked at what sort of genotypes gives you this sort of AD or neutralization antibody titer. So we've used our cohort in Vietnam, and this consists of 1,000 over participants with primary and secondary infection. And then we went on and we looked at the neutralization antibody titers. And we found that the PET value, the peak enhancement titer, is consistently higher in secondary infection as compared to that of primary infection. So we think that this particular PET titer would be useful for differentiating between these two infections. That's one. And also, there is a possibility of when you have secondary infection, there will be much more higher levels of antibodies that neutralizes the virus, which in fact gives you this higher PET value. So when we did a neutralization titration with a cells that do not express the Fc gamma receptor, this was not as accurate as the PET titer which we were doing. So, and of course, we then move on as to look at what sort of antibodies which are actually involved in giving you all this neutralization and infection enhancement activity. So if we look at the Igg genes, it consists of a whole host of reassortments from different genes, which is the V, D, J, and C gene. So this reassortment of the genes gives you 10 to the power 17 different kinds of Igg immunoglobulins, which are involved in the interplay of protection towards dengue. So here we can see that there are multiple combinations, thousands and billions of combinations of this IgG arising from the BCR gene itself. So what we are actually interested in is to tease out what sort of BCRs are involved when you have a secondary or when you have a primary infection, or when you are when you have recovered from that, what sort of antibodies which are giving you this certain sort of protection, or let's say neutralization. So in this case, again, we've used the cohort from Vietnam, and we took samples, serially collected samples from our long-term cohort to look at what sort of differential BCR expression which are specific towards dengue. So in this case, we did a BCR IgG repertoire, and we compared between primary and secondary infection and also against healthy controls. Well, interestingly, we did identify several variable regions which are which we think is involved during the dengue convalescent samples in dengue convalescent phase. So, but we really look at the clones when we look at the IgG and IgM clones and the clonality between all the patients. We actually did not find any shared sequence between two or more patients, specifically on the IgG and the IgM. So we think that this is probably based on more on the convergence of the BCR itself. Does not necessarily gives you this certain sequences which are similar across all dengue patients, but when all these multitudes of BCRs which are unique, which are some sort of a barcode for each of the patients, are combined together, they will give an convergence of these antibodies, which will result in certain activity, neutralization, or ADE against dengue. So then when we look back, it's probably not really related to, it's probably BCR is also important, but we went on to look at the TCRs, the T cell receptor sequences, and we went on also to look at the HLA, to look at the antigen presentation, those sequences which are involved in the antigen presentation between the B cells and the T cells. So this is called the TB cell collaboration. So we look at the T cell exposed motif in the dengue patients, and we found that there are several motifs. These are the top 14 motifs which are mapped to dengue virus itself, and they are shared across most of the patients. So this was not seen in the BCR itself, but this is probably something which drives the uniqueness of this dengue antibodies, which leads to recognition of the virus itself. So we were able to map what sort of, whether it's structural or non-structural protein, which is involved in the antigen presentation of the virus itself, not just to the dengue 1, 2 or 3, it's across all serotypes. Some patients gives you, again, this is the results from just dengue 1 patients, and the majority of them has a T cell exposed motif, which is reactive towards dengue 1. But there are several patients across which shares similar motifs that is reactive across dengue serotypes. There are also some patients which also recognizes the West Nile and the yellow fever virus. So this is a algorithm-based calculation. And then we went on to calculate what sort of, how many percentage of these, of how strong the binding will be. And we went on to synthesize all these peptides, which were mapped to the virus itself. And we look at the stimulatory of the IL-4, and we found that these peptides do stimulate the IL-4 of the PBMCs. So we think that what is identified over here is useful in the identification of what sort of antigen sequences, what sort of epitope is presented for the T cells. And with the help of that B cells, antigen presentation of the B cells, that is where you get a much more stronger response towards the virus itself. So the work is still ongoing on this, how the interplay between different flavivirus infection gives you this sort of immune responses. And we've identified several candidate motifs that are associated with primary and secondary infection. We have also identified several T cell motifs, and the degree between sharing of each motifs, there are some motifs which are shared across all dengue patients which we have tested. And we're most likely going to accumulate more data using much more dengue patients. And with that, we hope that this understanding will be able to accelerate our understanding towards how all this cross-reactive immunity gives you this sort of dengue presentation, or does it give you protection against the disease? So moving forward, we are working with our counterparts from Myanmar, Philippines, Malaysia, Nepal, and Sri Lanka to look at what sort of dengue virus outbreaks is going on. And it's important to monitor what sort of arbovirus outbreak is going on, because all this interplay will, in the end, affect the epidemic patterns of the region, epidemic landscape of the region. So we also went on to look at development of assays, which may help in looking at how these are diagnostics, perhaps, and which looks at how the how high the antibody titers are. And we are also looking into much more rapid assays, which will be able to tell you what sort of IgGs you have, what are the levels of IgG you have against each of the different arboviruses. Where we're also looking at the mechanisms behind that, because the total activity will, the activity in all will tell you what exactly the titer is, whether you have neutralization or whether you have AD antibodies, but it does not exactly tell you what's happening behind that and what's driving all these mechanisms to give you this certain immune response. So we are also interested into looking into that, and also using our mouse and animal model, which is the non-human primate marmoset model. I did not have time to share it with you over here, but this is an animal model which is useful for vaccine therapeutics as well as studies of mechanism. So I would like to acknowledge all our colleagues, which was all our collaborators, which were very helpful in this project, and especially from our counterparts from Vietnam, and also from Nagasaki University, and also from WHO. So, okay, with that, I would like to end the presentation. Thank you.